Hi friends, welcome back to my channel Only Bio. Presenting up the important points for the chapter cell division from 101 to 120, useful for different competitive exams, and yes, it consists of many tougher points in this particular segment. Point number 101, a network of filament that forms mitotic and meiotic spindle during cell division is termed as spindle fiber. Movement of chromosomes during mitosis and meiosis cell division is due to the spindle fibers. Next, spindle fibers are made up of tubulin proteins and RNA. It is composed of microtubules, which is a polymer of alpha and beta tubulin dimer. Next, spindle fibers along with associated proteins, condensed chromosomes and centrosomes or asters form the spindle apparatus. Next, spindle fibers get attached to kinetochores and arms of chromosomes and help in segregation and movement of chromatids. Next, the next point, many cells continuously divide, such as the cells that line the human digestive tract or the cells that make up the human skin. Next, cell division is stimulated by certain kinds of chemical compounds. Molecules called cytokines are secreted by some cells to stimulate the others to begin the cell division process. Next, also contact with adjacent cells can control cell division. The phenomena of contact inhibition is a process where the physical contact between the neighboring cells prevents cell division from occurring. When contact is interrupted, however, cell division is stimulated to close the gap between the cells. Point number 108. Cancer is potentially a deadly form of uncontrolled cell division. Next, cells undergoing division are also termed as competent cells. Next, the time G1 lasts varies greatly among eukaryotic cells of different species and from one tissue to another in the same organism. Next, tissues that require fast cellular renovation, such as mucosa and the endometrial epithelia, have shorter G1 periods than those tissues that do not require frequent renovation or repair, such as muscles or connective tissues. Next, the cell cycle is highly regulated by several enzymes, proteins, and cytokines in each of its phases in order to ensure that the resulting daughter cells receive the appropriate amount of genetic information originally present in the parental cell. Point number 113. Cells are mainly induced into proliferation by growth factors or hormones that occupy specific receptors on the surface of the cell membrane, being also known as the extracellular ligands. Examples of growth factors are as such: epidermal growth factor, in short called as EGF, fibroblastic growth factor, in short called as FGF, platelet derived growth factor, in short called as PDGF, insulin-like growth factor called as IGF, or by hormones. Next, PDGF, that is platelet derived growth factor, and FGF, that is fibroblastic growth factor, act by regulating the phase G2 of the cell cycle and during mitosis. After mitosis, they act again, stimulating the daughter cells to grow, thus leading them from G0 to G1. Therefore, FGF and PDGF are also termed competence factors, whereas EGF and IGF are termed progression factors because they keep the process of cellular progression to mitosis going on. Point number 115. Growth factors are also classified along with other molecules that promote the cell cycle as pro-mitotic signals. Hormones are also pro-mitotic signals. For example, thyrotropic hormone, one of the hormones produced by the pituitary gland, induces the proliferation of thyroid gland's cells. Another pituitary hormone known as growth hormone or somatotrophic hormone, that is STH, is responsible for body growth during childhood and early adolescence, inducing the lengthening of the long bones and protein synthesis. Next, estrogens are hormones that do not occupy a membrane receptor but instead they penetrate the cell and the nucleus, binding directly to specific sites in the DNA, thus inducing the cell cycle. Next point, anti-mitotic signals may have several different origins, such as cell-to-cell adhesion, factors of adhesion to the extracellular matrix, or soluble factors such as TGF-beta, that is tumor growth factor beta, which inhibits abnormal cell proliferation, proteins such as P53, P16, P21, etc. These molecules are the products of a class of genes called as tumor suppressor genes. Next point, oncogenes, until recently, which were also known as proto-oncogenes, synthesize proteins that enhance the stimuli started by growth factors, amplifying the mitotic signal to the nucleus and or promoting the accomplishment of a necessary step of the cell cycle. Next point, when each phase of the cell cycle is completed, the proteins involved in that phase are degraded so that once the next phase starts, the cell is unable to go back to the previous one. The last point, point 120, next to the end of phase G1, the cycle is paused by tumor suppressor gene products to allow verification and repair of DNA damage. When DNA damage is not repairable, these genes stimulate other intracellular pathways that induce the cell into suicide or apoptosis, which is also known as programmed cell death. Friends, hope this is helping you out, and please share this up with maximum friends and keep studying with me. Thank you so